This is an immunoglobulin G antibody. It specifically is immunoglobulin G1 antibody, which you can tell based, in part at least, on this double disulfide bond. So in this antibody and every other antibody in the human body, you have a heavy chain, which is the dark twist tie on both sides, and a light chain, which is the white twist tie on both sides. The main difference is the mass, and basically the amount of amino acids in each of these. So the heavy chain has more aminos, and the light chain has less. Each little segment of these sheets are comprised of beta sheets. So I have a little zoomed in version. So this is my zoomed in piece of, let's say, this right here, the light chain highlighted in red. So when you zoom in on each of these components, light chain has 1, 2, 3, 4, heavy chain has 1, 2, 3, 4, 5, 6, 7, 8. So here you have a beta sheet. One sheet is red, one sheet is yellow. They're bonded in the middle with this little black line. That's a disulfide bond. And I'll explain the little squiggly lines later. Antibodies have a constant region, which in my case is the unhighlighted area. So anything not highlighted in red is a constant region. And these are the same from antibody to antibody. Doesn't matter which B cell made them. The variable region is in red, highlighted in red. These are a result of the recombination of B cell genes, and they makes for a unique variable region on the antibody, where the antigen is going to bind, which is going to be in this area. The antigen site up here can be kind of a groove. It can be just sort of an open face or an extended surface. It can be a pocket or something that will kind of go over the antigen. So a knob is kind of what the textbook calls it. And this site is configured for a very specific ligand. So that can be something that's on the surface of an entire pathogen, or a fragment of that pathogen, or some other antigenic epitope. Next you have the hinge region, which is the wire that's going down through this heavy chain. So the hinge is part of the heavy chain. The hinge is what gives an antibody its flexibility. Immunoglobulin G is particularly flexible. It can rotate, which is going to be rotation around this axis here. It can wave, which means these two pieces can kind of wag back and forth like this, let's say, like this. And then this bottom piece can also wag back and forth. And then you have kind of an elbow system here as well, which I could show you since it's a twist tie, like that. Okay, and then lastly we have the FC effector region. So this is the FC piece from here to here. This piece is what's going to interact with the human cell, with the effector cell usually. So if the antibody is soluble, this is the region that's going to bind to whatever effector cell is going to interact with the antibody. For example, a macrophage that is engulfing a pathogen that's been opsonized by antibodies will interact with this piece. If your antibody's membrane bound, this is going to be the transmembrane section, the FC section, usually this bottom set of beta sheets. Let's see, what else can I tell you? Oh, immunoglobulin G is a monomer, so it's just going to operate in its one singular form, as opposed to being a dimer or a pentamer. which means those antibodies require multiple copies in order to be effective. What else? Oh yes. We'll come back to this here briefly. Told you I'd explain what these squiggly lines are. Squiggly lines are highly variable regions. So if this piece is this piece, you can see these three dark blue or black regions are going to be highly specific to whatever antigen this antibody is going to bind. Okay, I think that's everything. Thank you.